Good day, viewers. This is 300 Plus Academy, where all we do is exams tutoring, ensuring that you ace your next and, of course, every exam. Moving straight into the business of the day, it's about 2024 NECO SSC exam. This time around, we'll be looking at biology practical. As far as this practical is concerned, we've been given about 15 specimens, which you can see on your screen. And after careful examination, examination of these specimens, we've been able to group them into four, because we can see certain relationships, in there. So we have about four groups. So we're going to take each groups in a separate lecture video. So for the very first group, talking about onion bulb, ginger rhizome, Irish potato, as you can see on your screen. So what are the probable questions? Talking about the very first group, that's specimen A, B, C. The question says, identify specimen A, B, C without reason. In this case, you don't have to give any reasons. So the answer is quite simple. It is just for you to list. A is onion bulb, B is ginger rhizome, and C is Irish potato. That ends question, the first question. Question 2 states, classify specimen A, B, and C into its division and class. When we look at specimen A, B, and C, as you can see, they are all tracheophyte. And when we say tracheophyte, it simply means that these are plants with vascular bundles, and that has been lignified, they've been hardened. Yes, that's what that means. They just basically have vascular bundle. That is why the division. Recall that when we look at things, we have kingdom. When we want to classify based on taxonomy, kingdom, phylum, we use phylum for animals, but more specifically when dealing with plants, we use the word division. So when you look at specimen A, B, and C, they are basically tracheophyte. That's we say tracheophyta, simply because they have vascular bundles for conduction of water and manufactured food. Now, talking about the class of specimen A, B, C. Specimen A and B are both Liliopsida. You can see that Liliopsida. And white specimen C is actually Magnoliopsida. What that simply means is that A and B are monocots. They have a single cotyledon, whereas for specimen C, talking about Irish potato, it is a dicote, and that's what we call Magnoliopsida. So you can see that in just in case you have that question. So that ends that. Moving on to the next question, 2B, we were told to state 3 features that we can see, observable features of specimen A, which are characteristic of the class mentioned. So the class we mentioned for A is the Liliopsida. So they're asking us, what are the features that we can see in A, observable features that are characteristics of Liliopsida. As you can see on the board, on the leaf, there is what we call parallel venation. Yes, you can see the diagram, what we mean by the veins on the leaf, the venation. So it is parallel for Liliopsida. That's exactly what you should notice about A. Recall that A is onion bulb. On that, for the onion bulb, on the shoots that you have, on the leaf that you have, it has parallel venation on the leaf. And then also, you can as well say presence of long, narrow leaves. Yes, these are features of the class Liliopsida. And also, presence of adventitious roots. You can see that in the diagram displaying on your screen, presence of adventitious roots. So these are observable features you should be able to notice about specimen A, which can actually be seen in the class Liliopsida. And that ends that question. Okay, moving on to the next question, question 2C. We were told to copy and complete a certain table, putting a tick where appropriate. Let's look at what the table looks like. We were told to actually put a tick where necessary here. Now, looking at this, is specimen A an annual crop? Is specimen B? Specimen A is onion bulb. Specimen B, ginger rhizome. Specimen C, Irish potato. Are they annual crop? No. So this is not necessary here. But when you look at the three of them, they are perennial. So when we say perennial, it means these are plants that persist, that remains for more than 2 years, for many seasons. They still continue to produce. But when you talk about annual crops, after just a year, once they produce, then they die off. So when you look at specimen A, B, and C, they are all perennial crop. So it is not necessary for us to put any markings on annual crop, because specimen A, B, and C are not annual crop. Now let's look at the three specimens. Are they food crop? Onion is a food crop, but specimen B and C, ginger and Irish potato, they are cash crop. These are crops that are basically grown for the purpose, of selling off. But in the case of onion, though commercially it is still being sold, but it is classified as a food crop, just for consumption. So that ends that. Let's look at it. Are they monocote or dicote? Just like we said for 2A. When you look at the specimen A and B, they are liliopsida, and what that means is that they are monocote, a single cotyledon. So I'm just going to mark this. It is necessary. But when you look at specimen C, Irish potato, it is a dicote. It is having two cotyledons. So that just ends the table. What we were told to do is to copy and complete the table, putting a tick or what we call a mark where necessary, and that just answers question 2C. So moving on to the next question, as you can see on your screen, we were told to identify the storage organs of ABC. Identify the type of storage organs of ABC. A is a bulb. B, that's talking about ginger, is a rhizome. And C is a tuber. That's a very direct question. We move straight to question 4. For question 4A, we were told to name the parts of the plant in which food is stored in each of A, B, and C. So for A, for B, for C, where is the food stored in each of these specimens? This is question 4A. In the case of specimen A, talking about our onion, food is actually stored in the leaves. As you can see in the diagram on your screen, food is stored in the leaves. And talking about B, food is actually stored for the rhizome. Food is stored on the stem. And for specimen C, food is stored in the tuber. Yes. So these are the storage organs of specimen A, B, and C, and that just about ends that. Moving on to question 4, we can have the question in another format. We were told in this question 4B that we should study specimens A, B, and C very well, and we should use that to copy and complete the table below. You can see the table. We were given the specimen. We were told to name the part modified to form those specimen. We were also told to state the mode of propagation and the materials stored in those specimen. In the case of specimen A, the part modified to form the onion bulb is actually the shoots, because you're going to see in the image on your screen, when you look at the onion bulb, you can see the knob-like compressed stem. That's what is modified to then form the onion. So that is why we say it's the for onion, it's the shoot that is being modified. In the case of a ginger, the stem runs horizontally, so it is the stem that is being modified to form ginger. In the case of a potato, that's Irish potato, it's going to, the potato itself is now going to be the root buried beneath the soil. So what is modified here is the root to be able to form specimen C. Look at it again. What part is modified to form the specimen? The part modified to form the onion bulb is actually the shoot. You have the adventitious root. If you look at the diagram, you have the adventitious root, you have the knob-like compressed stem, and that's the part modified, that's the shoot, alongside the fleshy leaves, to form the onion bulb. For ginger, it's a rhizome, it runs horizontally, it's the stem that is being modified. In the case of a potato, it's the root that is being modified to form that. Here, what are the mode of propagation? Here it is the bulb, lateral bulb. Specifically, it's the rhizome here, and the parts modified, the mode of propagation here is actually the root tuber. That if we cut into pieces, there is a part of the tuber here, which we call the eye of the specimen. It's going to actually produce, it's going to germinate. So it is the root tuber, the rhizome, and then the bulb here, that is the mode of propagation. What are the materials stored in this various specimen? In the case of onion, if you cut it, you're going to see water. So water is there, sucrose, sugar, which is a disaccharide, is there. In the case of ginger, water is also there, starch is there, it's a plant, you understand. So normally glucose is synthesized, is converted into starch for storage. In the case of potato, starch or sugar are the stored materials. And that ends question 4b. So what are the differences that we can observe structurally between A and B? In the case of A, that's talking about onion, there are no nodes and internodes. Nodes and internodes are absent, as you can see in the two diagrams on your screen talking about onion and ginger. In the case of the ginger, nodes and internodes are present. Then when you look at the onion, the stem is knob-like, whereas for ginger, the stem is horizontal. You understand? The stem runs horizontally. Those are the two conspicuous structural differences. Moving on to question 5 now. We were told to state similarities between A and B. Look at it. The way they put it is that name the 3 parts common to A and B. So the 3 parts common to A and B is like saying the similarities between A and B. Terminal board present in both. That's why the diagram is displaying on your screen. Terminal board present in both. Adventitious root present in both. Lateral buds present in both. Leaves on the outside are reduced to dry scales. That's the outermost layer. You can see it's going to be a brownish, pinkish scale in the case of onion, and in the ginger, it's a brownish scale. That is the outermost layer. That's the leaves on the outside are reduced to dry scale. So those are the similarities, or what we say the 3 parts common to A and B. We've listed 4. You just can pick about any 3. So that ends question 5. For question 6, as you can see on your screen, it says suggest one method of propagation of A, B, and C. Suggest one method of propagation of A, B, and C. Now, the method of propagation, it is still there on the board. So you can just have it as a direct question, but we've answered, this table already talks about it. One method of propagation for A, B, and C, although all are can be vegetatively propagated, but A, it is the bulb, then B, it is the rhizome, then C, root tubers, that's the mode of propagation by the root tubers. That ends question 6. So moving on to question 7, as you can see on your screen, we were told to examine specimen A, B, and C and comment on their features of biological interest. So talking about the onion, what is of biological interest here? You talk about the shoots. You see, I said it has swollen on the ground compressed shoots or buds with inner scale leaves. Yes, the leaves, the scale leaves, as you can see in the diagram on your screen, the scale leaves are fleshy. The scale leaves are inner scale leaves which are fleshy and swollen with food reserve. You can see, so the shoots immediately after the knob-like stem, you see, are swollen on the ground compressed shoots or buds with inner scale leaves which are fleshy and swollen with food reserve. Now, a parent bulb, you know, the bulb as you can see on your screen, the bulb, let's say this is the bulb, and these are the fleshy, you know, leaves. This is the bulb here, is the bulb. So a parent bulb produces one or more bulb which breaks away. The leaves, now you see in the diagram on your screen, the compressed knob-like stem, you see. The leaves of the terminal bud grows up to manufacture food. That's the leaves of the terminal bud is the one growing at the center, going upwards. It's also in the diagram on your screen. And then you see the leaves, a parent bulb will produce one or more bulb which breaks away to form, of course, another onion bulb. And that is why when you see your onions, those type of onions that they are in twos, you see when you have a certain onion, the next onion is going to be growing just out by the side, which means a new bulb has been formed. So a parent bulb produces one or more bulb which breaks away. The leaves of the terminal bud usually will grow upward. You can see in the diagram on your screen. The leaves of the terminal bud grow up to manufacture food via photosynthesis for the formation of a new lateral bud. That's the side bud, and that's just about for the formation of a new lateral bulb, and that just about form that, so that the onion grows, you know, by the side, talking about a parent bulb forming one or more bulb which breaks away. So that is what is of biological interest as far as A is concerned. Talking about the biological interest of specimen, when we say biological interest here, you know, it just means we should comment on something that interests us biologically, you know, about that particular specimen. And when you talk about something of biological interest, it could be the way they manufacture their food, it could be the way they reproduce. And if you look at specimen A, B, and C, since they are all organs of vegetative propagation, I think the biological interest should be somewhat focused on how, you know, they produce, on how they propagate. The focus should be on how they propagate or reproduce. So when you talk about specimen B, ginger rhizome, as you can see in the diagram on your screen, it's a thick fleshy underground stem which runs more or less horizontally. You can see that beneath the soil. Some food manufactured is sent to the lateral buds. Look at the diagram very well. It's displaying on your screen. You see the terminal bud is there. You know, it manufactures food via photosynthesis. Now, some of the food is going to be sent to the lateral bud, and that lateral bud continues to grow, you know, the rhizome. You can see lateral bud which continues to the growth of the rhizome. The terminal bud, you know, that one is growing into the aerial shoot. So some food manufactured is sent to the lateral bud and the remaining one stored. Now, when the older part of the rhizome die, the lateral buds continue to grow. You understand? They continue to grow horizontally until they then give rise to another aerial shoot. That is what is of biological interest as far as specimen B, ginger rhizome, is concerned. Now, talking about the biological interest of specimen C, that's Irish potato. Now, it's a tuber actually, and there is a part here of this specimen which we call the axillary bud, alternatively called the eye. You can see that on the diagram. if we cut that specimen into pieces such that that piece has axillary bud or what we call the eye, and we plant it, that axillary bud is going to grow adventitious root. That's the eye is going to grow adventitious root, and it will also grow aerial shoots. It is the aerial shoot that manufactures food by photosynthesis. But what is of very important biological interest here is the eye or the axillary bud, which is during reproduction such that a new tuber is to be formed. It is the axillary bud here that is going to grow new adventitious root and is going to give rise to a new aerial shoot. Look at the diagram very well. You're going to see the axillary bud, otherwise known as the eye of specimen C. You can see axillary bud, that's the eye. If we cut that specimen into pieces, it is the axillary bud that is going to grow into a new aerial shoot so that it can manufacture food needed for the growth of the tuber. So those are the biological interests of specimen A, B, and C, and that just about ends question 7. So moving on to the next question, question 8. We were told to observe specimen B and C carefully. B and C carefully, and states 3 observable features of biological importance in this specimen. Look at it again. Observe specimen B and C carefully, and state 3 observable features of biological importance. In question 7, we were told to comment. When you are told to comment, it means, you know, just like a write-up, like we did for question 7. But when we are told to state, look at the way they presented the question. Observe carefully specimen B and C and state 3 observable features that are of biological importance. If you look at specimen B, what are of biological importance here? Foliage leaves. That's talking about the rhizome. You can see the foliage leaves in the diagram that grows upward. The terminal buds, the lateral buds that continues the growth of the rhizome, the brown scale leaves, adventitious roots, then the fact that they have nodes and internodes. These are of biological importance to this particular specimen. Here we were told to state, so we're going to observe the specimen and state those things that are of biological importance. If you look at specimen C, without doubt, the axillary bud, you know, is always of great biological importance because it is what is responsible for vegetative propagation of this particular specimen. You see specimen B, swollen stem, or you call it a stem tuber. It is swollen, as you can see naturally. Axillary bud or the eye, it has lenticels, scale leaves, and it also has adventitious roots. So these are various parts of the specimens that are of biological importance. If we were told to have a state for specimen A, talking about the onion bulb, it also has adventitious roots. You talk about the bulb, which is supposed to form a new bulb, and you talk of the terminal bud, which grows into leaves, photosynthesize, food is then sent. You talk of the lateral bulb as well. So that's just about things that are of biological importance there. We were told to state. So when we were told to state, we just state. Still on question 8, we were asked another question. It says, what is the relationship between B and C? What is the relationship between specimen B and C? The relationship is very simple. Both are modified underground plant stems that serves as storage organs. In fact, A, B, and C, they are just organs of vegetative propagation. But when you look at specimen B and C, the relationship between them is that both are underground plant stems. You see the rhizome is always on the ground. you talk about the Irish potato itself, is going to be underground. So they are both modified underground plant stems that serves as what? Storage organs, so that when the terminal bud and the leaf in both cases photosynthesize, food are sent to them for storage. So they are just modified underground plant stems that serves as storage organs. And that ends that question. Moving on to the next question. As you can see on your screen, we were told to state the type of reproduction common to all of specimen A, B, and C. Well, the type of reproduction common to them is asexual, or what we call vegetative propagation. And in that question, we were told to give one reason for each, one reason for thinking A, B, and C, can reproduce asexually or via vegetative propagation. So, what will be my reason for thinking that specimen A, onion bulb, can reproduce asexually or via vegetative propagation? Now, my reason is going to be because it has lateral bulb, which is capable of breaking away to form independent bulb. Remember that I said previously that if you look at, as you can see in the structure displayed on your screen, bulb, the compressed bulb there. You see, it's going to give rise to another lateral bulb, which breaks away to form an independent bulb, and that just about answers that. Talking about specimen B, why do I think it can reproduce asexually or via vegetative propagation? It is the presence of terminal and lateral bud. Remember that the terminal bud in a ginger rhizome, it is to manufacture food via photosynthesis, while the lateral bud ensures that the rhizome continues to grow horizontally, so that when the old rhizome dies, the new rhizome, the lateral bud continues the growth, and we have a new rhizome. So why I think B can reproduce asexually or via vegetative propagation is due to the presence of the terminal, more specifically the lateral. But when you hear the word lateral, it simply means by the side. So you see, for a rhizome, it continues to grow horizontally. Then for C, which is the most conspicuous, why do I think it can reproduce vegetatively or asexually? It is the presence of the eye, or the axillary bud, which if we cut the specimen and plant it, it is capable of forming adventitious roots from the eye, and of course a new aerial shoot from the eye. And that just about answers that. And with that, we've come to the end of the first part of this four lecture series, talking about NECO 2024 probable question for biology practical. For myself and the entire team, it's bye for now.